He works in the laboratory of Professor Joanna Ivaska, who is well known for her and their research on adhesion molecules, adhesion signaling, migration, invasion, and so forth. And this is the topic also of Aleksi. He's working on mechanotransduction mechanisms, and he has many interest in this respect related to especially cancer cells, interactions of the actin cytoskeleton with adhesion molecules, adhesion signaling, extracellular matrix, and inside out, outside in signaling. And I think with that, I'll leave the introduction rather short. And Aleksi, welcome to the pub, and over to you.
Thank you, John, for the kind introduction, and also thank you for the other organizers for giving me a chance to present some of our work here today. As mentioned, I work as a PhD student in Professor Ivaska's laboratory at Turku Bioscience, and our group is mainly interested in how the cells of a multicellular organism interact with their immediate physical environment, other cells, but mainly the extracellular matrix, and how the cells can interpret the biochemical and sometimes physical signals of this environment using what is rather complex molecular machinery to then direct their own behavior, including growth and movement. Integrins are some of the key mediators of these cell matrix interactions, and these are transmembrane proteins that, together with numerous signaling and scaffolding proteins, help connect the extracellular matrix to the intracellular cytoskeleton, typically actin, and then mediate both mechanical forces as well as biochemical signals. The integrins themselves are heterodimers comprising alpha and beta subunits, which both have pretty large multi-domain extracellular portions, single-pass transmembrane domains, and typically small intracellular domains. And the different subunits come in many flavors, and together they comprise more than 20 different integrin heterodimers, which have very different binding specificities for different matrix components, different binding kinetics, and also expression profiles. So all of this allows the cells to really adapt their response to the environmental cues. Now, in order for the integrins to actually bind their ligands, they have to be active. And this can happen by so-called inside-out signaling, where adapter proteins and activator proteins bind the intracellular tail of the integrin, which, at least with some integrins, leads to rather large conformational changes. So the bent extracellular domain of the integrin swings out and opens. And in this conformation, it has higher affinity for the extracellular ligands. These ligand-bound integrins can cluster together, recruit additional molecules to form larger adhesions, and in this fashion, they then signal inside the cell by various pathways, and this is known as outside-in signaling. There are different types of adhesions, from these transient nascent adhesions at the leading edge of the cell to focal adhesions, which are bound to actin trace fibers, are a more long-term solution for the cell to move around. And also in the case of stromal fibroblasts, the fibrillar adhesions that are important for matrix deposition and remodeling. And in these fibrillar adhesions, active alpha-5 beta-1 integrin swaps its original adapter molecule and activator tailing enzymes to remain active, and localizes in these large elongated central adhesions in the cell, which then grab soluble fibroactin and apply forces on those, allowing the fibroactin to form fibrils. And this fibrinogenesis creates a scaffold that then allows the deposition of other matrix components like collagens and kinesin C, promotes cell movement, and is critical for matrix remodeling. But we were interested in knowing whether these structures are actually responsive to mechanical cues. And this is biologically important because the cells in the human body, for example, encounter these types of cues all the time, from shear stress in vasculature to tensile forces, compressive forces. But maybe the most ubiquitous mechanical cue would be the substrate stiffness, because all the different tissues in the human body have their own intrinsic stiffness values or elasticity, elastic modulus. And these are also biologically important, and even in vitro cell types derived from many of these tissues respond strongly to the matrix stiffness. Integrins are used by the cell to sense these changes and differences in substrate stiffness. So what typically happens in most cell types is that upon stiffer substrates, and when the cell is exerting more forces itself by actomyosin, the integrin bonds are stabilized, integrins are clustering together to form stable adhesions, and actin is also organized to transmit more forces. And this leads to some signaling responses inside the cell, like regulation of megasensitive transcription factors. And this is all very well appreciated and known, but what has been less clear is whether the central fibrillar adhesions that are responsible for the matrix remodeling are really sensitive to forces and substrate stiffness in the same fashion. And in order to study this, we wanted to create a system for applying stiffness gradients on the cells while they are in culture. And we did this by combining two different acrylamide solutions, allowing the solutions to polymerize and mix together under a glass cover slip. And the stiffer of these pre-polymer solutions is also supplemented with fluorescent marker beads. And we then measured the density of these marker beads using regular confocal microscopy. This was an important point for us because we wanted to make this applicable for many laboratories and in many contexts. And then we used atomic force microscopy to probe the same regions and correlate the actual elastic moduli to the bead density. And what we got was this standard curve, which basically allows the estimation of the elastic modulus on the substrate without actually doing AFM during the experiments. So simply from the confocal data and the bead density, we can deduce the elastic modulus. And these gels were then functionalized with fibronectin before they were used for culture. Now, when we started looking at the adhesions themselves, we wanted to use turf microscopy whenever possible, because this allows us to get rid of a lot of the cytoplasmic backgrounding. We can really focus on the components of the adhesions themselves at the cell matrix interface. And in order to study the fibrillar adhesions, we first had to decide on the optimal marker to tell which adhesions we should be looking for. And there are different components that are known to be enriching in fibrillar adhesions. So we started by staining for different adhesion components, also for ones that are known to exist mainly in more peripheral adhesions, as well as fibronectin, and doing a radiometric analysis to compare their distribution to one another. And we noticed quite quickly that the tensins that are typically used as markers for fibrillar adhesions also appeared quite a bit in the more peripheral adhesions, whereas, for example, alpha-5 beta-1 integrin did not. It still co-localized very well with these fibronectin fibers, central parts of the cell, so we decided to use this as our readout for the fibrillar adhesions. And we were using telomerase immortalized fibroblasts in this study. And then we started putting these cells on the hydrogels, and we noticed quite quickly that indeed the alpha-5 beta-1 positive fibrillar adhesions were responsive to substrate stiffness. They elongated pretty quickly in the 1 to 7 kilopascal region before plateauing off, reaching the maximal size of just under 4 micrometers. And since tensins are known to retain integrin activation of they swap their adapters from tail tensins, we're quite happy to see that this is also relevant for the mechasensitive elongation of the adhesion. So if we deplete tensin 1 from the cells, The fiber adhesions elongate slower as a function of stiffness, but also they never reach their maximum size that they would be expected to reach otherwise. So we concluded from these results that the elongation of fiber adhesions in fibroblasts is dependent on both TLC1, but also importantly on substrate stiffness, hinting at the possibility of mechanical feedback in matrix remodeling, and this obviously has some potential implications for tissue fibrosis and cancer. But we're also happy that we managed to set up a simple and cost-effective tool for preparing the thickness gradient for cell culture, and importantly, they were also compatible with high-resolution imaging. Now I'm going to switch gears a bit and talk more about cancer mechanobiology and cell migration. The biophysical features of malignant tumors are in many ways exceptional from regular tissues, both in terms of microarchitecture and bulk stiffness of the tumor, and the compaction of the tumors. And all of this is intricately linked to the other biological aspects or hallmarks of cancer. One of the ways in which stiffness can direct cancer cell behavior is by influencing migration. Curataxis is the ability of cells to detect and migrate along stiffness gradients, and in almost all the recorded contexts for adherent cells, this seems to happen from soft, stiff environments. But this has been a bit of a paradox for cancer cell biology, because in many cases the tumors are much stiffer than the surrounding tissues, and nevertheless the tumors also are able to invade to the healthy stroma and form metastasis. So how universal is this positive conventional curataxis for cancer cells? For example, the breast carcinoma MDA MB221 cells have been recorded to go conventional curataxis. And fair enough, when we put these cells on our stiffness gradients and culture them for a few days, we see that the cells tend to cluster in the regions of higher stiffness, so all the way up to the 20 kilopascal mark. But there are also exceptions. For example, these U251 glioblastoma cells. So if we put them on the same gradients, we notice quite quickly that the cells actually seem to prefer regions of intermediate stiffness, and they are actively avoiding the stiffest region. This is quantified on the right. So this hints at the possibility that the cells are actually capable of migrating down the gradients, at least from the very stiff regions to areas of lower stiffness. So they would be capable of negative curataxis. But if we want to know if this is really going on, we need to take a step back and think about how these cells are actually moving. And this is a movie from one of my colleagues in Turku. I'm sure many of you actually know Guillaume. And I really like the movie because it illustrates how the cell is physically interacting with its environment. Check this out again. We can see the individual matrix fibers bending and buckling when the cell is moving around. And the cell is actively applying traction to its substrate to move. And we can measure this traction using so-called traction force microscopy. And the idea is that the cells are grown on an elastic substrate that is fitted with fluorescent marker beads. And when the cell is pulling on the substrate, these beads are moving around. And we can record their positions and then remove the cell using detergent or trypsin, and see how much the beads were moving around because of the cell when the elastic substrate relaxes. This is how it can look like in practice. We can see here on the right the beads slightly moving before and after the cell has been on the substrate. And we can then use this information of bead movement to calculate the displacement field, that is the overall displacement of the substrate. And this is used finally to resolve the inverse problem, so to estimate the amount of forces that the cells would need to be exerting to deform the substrate in such fashion. And this is an ill-posed problem, which means there isn't only one correct solution for the problem at hand, but there are many ways of trying to get the best possible estimate and reduce the error, and we are using one of those in our studies. Now, as mentioned before, many cell types display this behavior where on different substrates, integrins are clustering together, forming mature adhesions, which allows higher force transmission, and as a result, most bacterial cell types actually display this type of behavior where the attraction forces exerted by the cells increase as a function of substrate stiffness until it reaches some type of plateau. And at one very high stiffnesses, we cannot even measure the forces anymore because the TFM is simply not applicable anymore. All we measure is noise. And this is also the case for the MDA-221s. So up until the 20 kilopascal mark, where our stiffness gradients also end, these cells display monotonically increasing traction forces. But interestingly, if we then take a look at the glioblastomas that were preferring the intermediate stiffness range of the gradient, these cells have a very distinct drop in their traction forces. And this peak traction force seems to take place in the range of the gradient we were using. So this poses an interesting idea that the cells are actually migrating towards where they can simply exert the most traction on the substrate, so where they are the most efficient at pulling on the substrate. And in order to study this further, we used a different kind of gel setup. So we prepared 4 responsive stiffness gradients from 8 to 15 kilopascals by using a photocleavable acrylamide cross-linker. And when we cultured the glioma cells on those substrates, we noticed not only that they were clustering once again on the softer regions, concordant with their stiffness optimum, but the cells at the interface were also preferentially migrating towards the softer side, seen here in the bottom, which indicated that the cells are actually actively undergoing negative transactions. We then started studying these cells in more detail and wondering what's going on in their biology to make this happen. And the first interesting observation would be that these cells don't really form mature adhesions. So contrary to the MDA-221s, for example, most of the adhesion components are simply localized in these peripheral ruffles, but they don't really seem to form proper mature focal adhesions, even on 60 kilopascal substrates. Focal adhesion formation and increased force transmission is also linked to the regulation of microsensitive transcription factors like YAP. And so we use immunofluorescence to study the localization of this protein. And in MDA-221s, upon increasing substrate stiffness, YAP is localizing the nucleus very nicely, as seen in the bottom. But the gliomas are very poor responsive to thickness, and there's barely any difference in the YAP localization. And also importantly, in all of these cases, we don't see any differences that would be specific for the 10 kilopascal region, above the 10 kilopascal region, that would set apart the region where the stiffness optimum and the peak traction forces lie. Okay, so what's going on? Well, one way in which we can explain these differences is the so-called molecular clutch model. This isn't exactly a new concept. It was first proposed in the late 80s by Tim Mitchison and Mark Kirshner, who were studying neurite elongation and growth cone dynamics. And the model basically attempts to explain the cytoskeletal and adhesion kinetic at the leading edge of the cell with purely mechanical terms. And this is all based on the fact that when enough forces are exerted on chemical bonds, the bonds start to break up much faster and dissociate much faster. And in absence of any kind of biomechanical feedback, it leads to 3 distinct regimes on different substrates or different substrate stiffnesses. In the first case, if the substrate is very stiff, individual clutches, so integrin, adapter, actin, ECM assemblies are broken down very quickly. And the cell cannot really build up any significant forces. And this is kind of like a person who would be slipping on ice trying to move around. On very soft substrates, the clutch lifetimes are much longer, but the substrate itself is so soft that it's very difficult for the cell to build up meaningful forces to actually migrate. And then between these two extremes, you have the stiffness optimum where the traction is the highest. These behaviors have been modeled extensively by Professor David Odie's lab at the University of Minnesota, Twin Cities. And they have designed so-called cell migration simulator that runs a stochastic simulation using real-life parameters, measured parameters. And as an output, you get different cellular features, including cell migration, as shown on the right. And we can also use this simulator to study the effect of motor clutch dynamics on rheotaxis. So we can place these in silico cells on a repeating gradient or substrate with soft and stiff regions, like the one we were using in the experiments. And we can then design these substrates so that the stiff and soft regions correspond to different parts of the traction curve of the cells in question. In this case, we're using two different substrates, one that is softer than the stiffness optimum or traction optimum, and here the cells would be expected to undergo positive rheotaxis. And conversely, on stiffer substrates, the traction optimum lies left from the substrate, and cells would be expected to undergo negative rheotaxis. And this is actually what happens. So within 12 in simulation hours or so, we see that the population starts differentiating, and there are more cells on the softer regions. And in the case of the softer substrate, we see the opposite. So the cells are actually undergoing positive rheotaxis and clustering in the deeper regions. So this was, excuse me. This was very interesting, but if the motor clutch dynamics are really driving the rheotaxis raises two additional questions. Can we influence this system in vitro in our glioma cells to influence the direction of the migration? And more importantly, can we actually somehow investigate whether this mechanism is applicable to other different cell types or biological contexts? So if this is just some quirk that is specific for the glioma cells. And prior work has indicated that perhaps a bit counterintuitively, decreasing the amount of intracellular contractility, the amount of motors, molecular motors available for the cell, increases the traction optimum or pushes the optimum to higher stiffness. And this is maybe easier to think about in terms of the cell having more grappling hooks or more clutches relative to the motors that it uses to move around. Then, if we manage to do this in vitro, we would also expect to push the stiffness optimum up and perhaps get rid of the negative rheotaxis and simply promote positive rheotaxis. And we tried this in the glioma cells, and we were using a ROCK inhibitor to decrease myosin 2 phosphorylation, meaning that we decreased the contractility of these cells. And we saw that when we put these cells on the stiffness gradients, the DMSO-treated control cells still cluster in regions of intermediate stiffness, but the ROCK inhibitor-treated cells actually start to undergo only positive rheotaxis. If we look at the cells in detail, we see that they are still happily moving around. Even with the ROCK inhibitor, they are sending out multiple protrusions before deciding which way to go. And if we quantify this migration in different gradient regions, we see that under the stiffness optimum of around 5 to 10 kPa, the cells, both the DMSO-treated cells and the ROCK inhibitor-treated cells, are actually undergoing conventional positive rheotaxis. But the situation changes above the stiffness optimum. So above the 10 kilopascal mark, control cells undergoing negative rheotaxis, migrating down the stiffness gradient, whereas this is completely abolished with the ROCK inhibitor, and if anything, the cells are actually migrating up the gradient still, so undergoing conventional rheotaxis. We then wanted to see whether this is specific for the ROCK inhibitor, if we can get similar results using other myosin 2 inhibition methods. And we decided to use the direct myosin 2 inhibitor flepistatin. And with intermediate or lower concentrations of the drug, we actually do see a similar trend. So the cells seem to be undergoing exclusively positive rheotaxis, whereas with higher concentrations, the trends seem to disappear altogether, and it can be that cells are simply not moving around very much, or that they are just losing all of their sensitivity to the substrate stiffness, but we haven't tested this in detail yet. All right, so I mentioned that the motor clutch dynamics, or the motor clutch model, predicts this B-phase extraction regime, but most cell types don't behave like this, and they actually exert higher forces and form mature adhesions on bigger substrates. So what's going on here? And this is actually dependent on specific adapter molecules. It's mostly thought to depend on kaolin unfolding and the recruitment of vinculin to the adhesions, where these components can reinforce the connection between integrins and actin and allows integrin clustering and increased forces. But prior work has also showed that if we target kaolin and remove most of it from cells, at least fibroblasts can be pushed back to a B-phase extraction regime. So what used to be a monotonic relationship between substrate stiffness and increasing traction forces now becomes a traction peak. And we wanted to see whether this would be possible with the MDA2 response. And we used a double knockdown of both kaolin isoforms to deplete most of kaolin from the cells. And we saw a marked reduction in the amount of focal adhesions formed by cells, which was encouraging. And we also saw that the attraction forces on the stiffer substrates, or at the higher stiffness range of the gradient cells, was influenced and was dropping relative to control cells. And when we then cultured these cells on the stiffness gradients, we noticed that they started to phenocopy the glioblastoma cells. So they actually started aggregating or clustering in the regions of intermediate stiffness instead of migrating all the way to the best areas. So I'd just like to leave you with this model to think about. So we think that what is going on here is that the motor clutch dynamics are directing the cell's ability to exert traction on the substrate. And in the basic form of the model, this means that cells on very stiff substrates struggle to exert enough force because of the constant slippage of the actin from the clutches. And the cells are actually migrating down the gradient towards their optimum stiffness, where they can exert more traction. In most cases, however, this is prevented by the mechanobiological feedback and the kaolin and vinculin-dependent adhesion maturation. And of course, this is where all the other experimental results about the importance of focal adhesion components and focal adhesion function for Dürr is coming. So this ties in pretty nicely with the existing literature as well. I'd like to thank all the people who have been the driving force behind these projects. Camila Guzman and Nuria Barber-Tres were mainly responsible for the stiffness gradients and pupil adhesion work. Also, Maria Georgia Doe was doing a lot of the tension experiments. For the Dürr project, Mathilde Mathieu, who's a postdoc in our lab, has been working on the actomazing inhibitor experiments. And this whole project has been a very close collaboration with the OD lab. This is a kind of an older image with many of the friends and then members of the lab missing. But one of the key players has been Jay Ho, a postdoc who's been doing a lot of the simulations. Also, Kevin Park and Mark DiStefano from the University of Minnesota have contributed their photoresponsive gradients. And of course, I'd like to thank everyone else who has been contributing to this project. Our collaborators in Washington, University of St. Louis and Xi'an, who have been working on modeling to show that the same mechanisms are actually applicable to renewable growth cones. And then, of course, I'd like to thank Johanna for being a great mentor and all the funding agencies. And thank you all for your